I think we'll go ahead and get started here. I know we might have a few other additional folks joining us coming in. For those that don't know me, I'm Carrie Dudek. I'm the director of the Life Science Tennessee Foundation's Life Science Mentor Program. So we are a statewide program that helps support early life science companies. So those that are in the therapeutic space, med devices, diagnostics, etc. Many that are rolling out from the university to translate their research. And we provide mentorship and resources and programming and try to get those companies in a better state that we're engaging with investors and raising capital and gaining traction toward commercialization. So with that, if there's anyone that's interested in that program, we are actively accepting applications. So that's a rolling admission, and we take companies throughout the year. So I encourage you to take a look, reach out to myself if that's something you're interested in. As we get started here today, first, just want to thank our host, Perry Medical College. We greatly appreciate you guys supporting the program and hosting us today. This is a fantastic space, and we're excited to be here. I also want to let folks know that next month's event, while I have you all at attention now, will be in Memphis. We will be at the University of Memphis for that one. And we will be joined by Jason Butcher from BSI, who's focusing on international markets and regulatory considerations for life science ventures. So we all hear about FDA a lot. So what are some of these other considerations that you might want to take into account when thinking about a broader market opportunity? With that, I'm excited to introduce today's speaker, Adam Haberle. So Adam is make sure I get this right, Adam, a senior director in clinical development at Nkarta currently, but he also comes with over 20 years experience across biotech and pharma, including roles at Vertex and Baxter and Amgen. He leads programs from early development through clinical stages and has a really strong track record in science. And we are extraordinarily happy to have him as one of our Life Science Tennessee mentors in our mentor network. So for those companies that come in, may have one-off meetings with Adam to provide just some general guidance. So today, Adam's going to be sharing insights on designing early R&D strategies that generate meaningful data and set you up for successful clinical translation. And some positive and maybe less positive stories from his own background and experience. So with that, Adam, I'll let you take it from here.
Thanks, Harry. So first of all, I want to start with an apology because I know it says right off the bat we talk early development, but the reality is you kind of have to talk about all development. So I'm going to skip around a bit. If you don't think about the end, starting, it's kind of already too late. So with that in mind, sort of the session, we're going to talk a little about the problem. The main thing I want to focus on really is the 4 programs that I've been involved in. So 2 were wildly successful, 2 are not. And I think in a way, you almost know more of your failures than you do from your successes. And so we'll focus a bit on that. What made them fail? Why didn't they work out? From those, we'll extract a little bit, and maybe we'll leave with a blueprint, sort of a little bit of a, what do you do next if you're interested in this type of thing. Everything is a scientific milestone, but there's some harsh realities when you're in drug development, especially when you're in venture capital, life science, or, a big pharma mentality. And that harsh reality is that people don't really care. And it's kind of rough to say no one cares, but no one cares. And so once you kind of take that in, accept that, start thinking about what do they care about, that's when you can kind of start to move forward. And so really what you talk about, as an early stage inventor, you might have a program, you're very academic in nature, and you care about all these things. But if you don't think about financing, if you don't think about the FDA or the partner all the way at the end, as I said before, you're already behind the 8 ball. And you really need to start working on that really all the way in the beginning. So early research is not technical validation, it's also trajectory design. So that's sort of the core thesis. And one of the main going on is you can't be everything you have in mind. So you have to start with your early evidence package. And that really focus on a couple areas. You know, we have to have that study focus on multiple groups. If you're in basic science, you still have to think about your population. You know, if you have an idea, what's the payer indication? How many patients are out there? Can you get them? And if you don't think about all this in advance, you know, right now I've got just a quick example. I'm involved in an advisory role, very low stage group. And we're putting together some venture capital decks. We're already starting to look around. The venture groups I've talked to, even though we are so early on, they're already starting to ask, what's your total addressable market? What's your payer? How do you figure out what we can charge for this individual therapy? And if you can't bring that forward, we're already talking about what does the phase 3 design look like and what does it cost? And we haven't even gotten tonight. We also want to evolve all the way down, what's your cost of goods and manufacturing? And if your audience is just for an SBIR, maybe you can push that down a little. But eventually you're gonna want real money. Eventually you're gonna want to get involved. And when that happens, you have to, be able to answer these questions. You have to survive diligence. And there's nothing angelic about any customers.
You have to
Some
subtract.
stories that are require language that share at all. They're not fun. They're not on your side. And there's a lot of early stage investors who end up with a non-voting board seat. Maybe 10% of the company, lack of control, and then pushed out by the time they can go out. And those are very real stories that happens a lot.
Already on the phone.
So I always like to be a little philosophical when I talk about drug development. And with that, the answer to every single question in drug development is the same. And the answer is it depends. And so you think about what you have. Startups, a lot of times, it's not one thing that takes them down. It's not one thing that delays them. It's a lot of things. It's a combination of things. They stack. Once one thing goes wrong, another goes wrong. And before you know it, you might have to catch the team. And so the timeline is usually set by the weakest link in a stack. And so just an example of a stack about shaping trajectory. You've got your molecule. Do you have the potency, durability? Can you manufacture it? There was another program I was involved with, which we had a great scientific story. We actually generated 48 molecules, small molecules. Not a single one of them worked in the assay. On paper, on a computer model, they all looked fantastic. It's all Schrodinger, Marcuse search. We really did hardcore medicinal chemistry. It just didn't happen. That's not what do you have a biomarker or an assay? Can you figure out a way to give a readout early and for less fun? Patient.
Oh, wait, hold on. To get there.
Who are you serving? Is there an unmet medical need? And that unmet medical need changes all the time. When I was at Baxter, we had licensed product for something called pancreatitis, a very rare form, or relatively rare form of autoimmune disease. Now, over the course of time, while we were putting all this money in the program, rituximab did some trials. Rituximab is a great drug. So rituximab did some trials in pancreatitis, and guess what? It worked. So we no longer have it. Now, these patients are still in a fantastic quality. It's a very difficult space. But there's no money to drive any program like that. Rituximab is a big benefit. And so you have to always look at continually assess things. Regulatory logic, can you get your, with the amount of money you have? That's very important. A lot of people seem to think that, and I'll give this with one of my examples, but a lot of people think that when you're in a big pharma, you have unlimited budget. It's actually not true at all. The same things you go through with venture capital, you go through a big pharma. You just have a different audience. You're not going to Deerfield or Atlas. You're going internal. But if you think they're any less hard on you, that's not true at all. I mean, they're massive. And while if your program gets cut in big pharma, you might end up with a different job program. So you're not worried about your job. Your program.
I go hop on this call.
So that said, common traps. You've got an elegant wrong question. That happens all the time. You think you've got all these wonderful things, but this gets back to the point of, does anyone actually care about the answers? Biomarkers, talked about that. And one of the programs that we get to, actually the first one is very much based around a biomarker. That's very, very predictive. And so that's an easy decision. Another one is about the assumed patients. Sometimes ultra rare diseases are very, very attractive from a financial point. You've got all kinds of things from a regulatory basis. FDA has these priority review vouchers. Talk about that with the second example, but those priority review vouchers are transferable. And they currently, when you're doing a venture capital calculation, they mark about $90 million right now. And so if you have those priority review vouchers, they allow someone to put that to the FDA and decrease their PDUFA time by 4 months. That can make a huge difference to a mature pharma. So someone like a Pfizer will buy it for $90 million. And they go on auction. They go anywhere from lows of like 50, 60 at a particular time to all the way up to about 300 at another particular time. So all these things kind of factor in common trust. So this is what we're going to talk about, these 4 different programs, 4 trajectories. And I really, on this one, I kind of based them on this graph. And did you have decision enabling data? And did you have registrational readiness? You know, one of these is from a very big fund. It's called Haberle. It's a cholesterol lowering drug I worked on when I was at Amgen. Another one is called Inhibrx 101. And this is something I was actually involved in this one, not directly. But when I was at Baxter, we looked at licensing it. And so I kind of saw the other side of that. Those are the 2 successful ones. And, you know, part of that success might be because they're in the upper right line. That's a very fine line. High signal rates, you know, more executional and registration rates. So this is where you want to be. If you think you might be in that bottom left quadrant, you might have some issues, might have some problems. 2 other ones I was involved in. One is called DOS 181, and that's with a small biotech company. That was a flu or antiviral potential. And ACG 801, which was recombinant human acid surroundings. And so with that, I'll start with Amgen. And Repatha is an LDL lowering drug. And so it comes into the field of cholesterol lowering with a lot of data already existing. So what does it do? Well, it lowers LDL. That's easy. Everyone knows that. Does lowering LDL lead to cardiovascular improvements and cardiovascular outcomes data? We already know that. You know, we've got all the data from all the statins, and there are, you know, 5 registered statins available. And so it's pretty much, when you talk about the FDA, it's considered settled science. So you already know that. The question is how strong it's going to be, and does it have any other, you know, potential issues. So when we ran the Repatha program, it's something called a PCSK9 inhibitor. So it's a different mechanism of action than a statin. But the main thing is during the phase 1 healthy adult blinded trials, we unblinded ourselves. It was so powerful that we saw 50, 60, 70% drops in LDL in patients before we even got out of phase 1, since we literally healthy adult trials that were running as fast as possible dose escalation. So right off the bat, you know, we had a causal disease logic that we had. We knew something about it to go forward. So at Amgen, and pretty much everyone has this idea, and at Amgen it's called MEWA. It's managing investment with opportunity. So that's sort of your venture capital moniker that you get in pharma. So it's very simple. It means how much, you know, in a brutal sense, is how much is this going to make us? If it's a lot, we're going to put in. If it's not, we're not going to bother. And so with a potential drug on the way that was going to be a antibody, so weekly injection, very high ability to charge for it because you're betting on the effects. So when you talk about, you know, where you're going, what you do with it, and you know where you're going, you know what it's going to bring you to the bottom of the So that's kind of the point. Amgen was playing major multi-points. And we had a standardized assay. LDLCs, easy. Everybody does it, right? Central lab, fast the patient, send it in, better output. So we had a portable story, an easy story. And while it's easy, you know, when you're working on drugs, it's easy to say, well, that's Amgen. My point really is it's not. This is the same way it works, even with venture capital putting in $5 million or venture capital or, you know, somebody looking to do $100 million Series B, or even someone taking 12 questions are the same. So it doesn't really matter. The story matters. The output matters. So we actually filed and got approval just based on the marker that drop in element at. But that's not enough. So the next question becomes, can you take that familiar marker and prove it really does? And that was a trial before, it was called an outcomes trial. So in an outcomes trial, you're not just looking, you're looking for an outcome. In this case, it's so-called MACE, which is a cardiovascular outcome. Heart attacks, it was secondary prevention. So the first part is you had to have had an event taken into the trial, had to have had a heart attack, had to have had a stroke, had to have had a stent replacement. So that already puts you in an increased risk because the single greatest predictor of a cardiovascular event is a cardiovascular event. And so that was your entry criteria. The original study was a 22,500 patient study, which was, substantial by any means. Our budget was 365 million for the trial. We're not playing around. This was in pretty much every country in the world that you could safely, even some not so safely, get in. We had a site in Beirut. I didn't go, but other people went. We're all over it. And so you go where you have to go. But at the same time, we were in a race with Sanofi. So Sanofi had a very similar, and I'll admit, pretty much identical product. They both PCSK9 inhibitors. So we actually did a analysis. This gets back to don't think of the amount of money spent. Think of the concept. How do you shorten time? And in this case, it was statistical. Because it's an outcome trial, we had a certain number of events. If we massively increase the size of the trial, we could take a year off the trial. So that's what happened. We went from 22,500 to 27,500. That's an increase of about, I think it was about $50 to $75 million spent. But it took an entire year off the trial. And if you're thinking about that, you're looking at a potential blockbuster. And you take your money off the end of patent life, which means your greatest peak years. So that's $1 billion. The potential pre-sales by taking a year off the trial. So that's, thinking about that. Is it worthwhile to spend $65 million? And the answer is it is. And so that worked out quite well. So let me pass the lessons for startups. Get back to the causality, right? Does your readout sit close to disease biology? LDLC? So it's easy. Settled science, not a big one. Do you have something like that? Probably not. That's a very unique case. But you might have other things that you can do. The second part is interpretable. You might not have the settled science as LDLC. You probably have interpretable. Can you tell a story? And people like to hear a good story. So don't get too far into the weeds. Don't think that, you're just going to present the data. You're just going to hit the science. People like a good story. And sooner or later, you're going to be presenting to a banker who doesn't know nearly as much about the history. Sometimes they don't know nothing, but they're your audience. Okay, it's for something to deal with. Part of it also is affordability. Can other individuals use that some is it set up as a difference? So this is where it gets the story. If your readout needs too much translation, it'll struggle making its use. Second example is a kind of fun one. By fun, I mean not fun at all. This goes back to one of the first jobs I had. So it was a company that was called NextBio at the time. This has been rebranded as Hanson Therapeutics. And it was a antiviral. At the time, it was for flu originally. But viruses enter the lung epithelium through something called the sialic acid receptor for sialic acid molecules. And this had a sialase. And so it would cleave that sialic acid. The idea being if the flu virus, if the other virus, whatever different virus can't bind, it can't bind, you cleave that at moiety, can't bind, it can't enter the cell, you don't allow infection. Flu is interesting because a lot of flu is based on reinfection. So you think, oh, well, it's already in there. That's not really it. The fluid has to reinfect. And that's also how Tamiflu works. A little bit different. Tamiflu is an aramidase inhibitor. So it actually stops the flu from budding off. But if you look at it, this is just overall time. I joined the company in 2006. So I filed the IND originally for that. Something should stand out to you in this. When I say I joined the company in 2006 and the latest public phase 3 target is 2025. It's not a good roadmap for a drug. Think about it, you have roughly 17, 18 years with continuation on a patent. None of the patents still exist on this drug. They're all off. And so, we started off early, and it was a very different model because this company was all based on SBIRs, contracts through NIAID, BAA, broad agency announcement contracts. They had quite a bit of money. They actually had close to $100 million of federal funds. And things happen. One of the things we got out of phase one, and this happened to be, I don't know if you'll remember the swine flu days, but we were all ready to go. Flu season always starts maybe October, so you want to be ready for flu season. Try to catch all in life. Swine flu came through in August. We absolutely did. And the question became from this is science. The reservoir, the viral vectors. Do you still have a reservoir after swine flu came through? Or are you going to have your regular flu season? Of course, because it was federal money and we had plenty. But we said, oh yeah, it's definitely going to happen. It didn't happen. And so we had swine flu. We didn't have a regular seasonal peak. So we set up all this, great big phase 2 trial in the US and Canada. And I think we enrolled about 23 patients at the time across 115 sites. That's not a good track record. And so that's things that can happen. Things get set up for failure. We did actually end up getting into the southern hemisphere for what was our winter, and did some good catch up in South Africa and Australia, but it wasn't enough. All the while you're burning cash. This was not a cheap product to them. So the company ended up not doing so well. I ended up leaving the company. I ended up moving on to Baxter, which was at the time a very good choice for me. And it kind of stagnated. There's some other reasons for stagnation. I've got a slide coming up that'll explain some of that. But the company kind of stagnated. Patents were still there until eventually rebranded. And it rebranded into a rare disease company. So that's the other thing. You're going for flu. Flu has major competition. That story for flu, it was all based on not venture capital, but who's your audience? It's based on government. It was based upon BARDA, which is, the Advanced Development Authority. It's the bioterror, something like group, Advanced Development Authority. But it's the group that was also behind all the moonshots with COVID vaccines. So this was in the early days. And they were worried about pandemic, bird flu, pandemic preparedness. So all the money going to this program came out of that. It had very little commercial appeal. So where do you go? So that's sort of the questions you have to ask. So at some point it got rebranded and went to parainfluenza virus in immune compromised patients. And so you've got an audience there. I don't know if you're familiar with, when people go through transplant, but the immune system has to be depleted. It's gone pretty much. And parainfluenza virus, you don't hear a lot about it because everybody here can clear it on their own. It's not fun. It's nasty cold, but you clear it on your own. If you're immune compromised, you can't. So what do? There are no drugs for it. Tamiflu doesn't work on that. So they actually found something that was fairly decent, and they did take it forward. But the problem is, by the time they took it forward, by the time they moved it ahead, it no longer had bad enough. So that's why we don't see it. Who's going to now take this and commercialize it? There are more costs than just proving something in clinical trials. It's easy to say, oh, I'm going to get the phase 2 clinical trial done. I'm going to show efficacy, and then someone's going to take it forward. The amount of cost that you have to do for a validated phase 3 for BLA ready, NDA ready commercial run from a CMC, chemistry manufacturing control. It's not, and it takes up to a year to get through it. You're talking about $50 million spent easy. So who's going to spend $50 million to get you something that has no patent life left? Maybe you've got some orphan disease. So in the US, you can say, I've got 7 years exclusivity. But that doesn't apply to Europe. Europe's a different orphan system. That doesn't apply to the rest of the world. When you did your patents, did you bother going into Asia? Some people did. So now you're limited. You say, I have 7 years exclusivity in the United States without a patent. And that gets back to the original story. Does anyone actually care about your story? In this case, the answer is no, they didn't. So it went away. So again, this is what I talked about earlier. respiratory viruses, a very large amount of them are solid gas. And so you had air influenza, respiratory syncytial, all kinds of things. And so they didn't have a focus. They had a very broad look. If they had come in early on and said, we're going to go early in the patent life, we're going to focus on something we can address. There's an unmet medical need. Parainfluenza, and immunocompromised individuals, parainfluenza often end up on ventilators. So now when you talk about price, how do you calculate the price you're going to charge? Well, I can now make some back calculations of hospitalization costs, what respirator costs, what death does. And it sounds really callous to say there's a cost of death, but there is a cost of death. They do count for that. And that's how you did figure out what you charge for your program. So you figure all that out. Maybe you get to the point where you're talking about, I could charge potentially $25,000 for a 2-week course of drug. Sounds like an absurd amount, but that's what you need to make that. Now, sometimes nothing can help you. And this is true. This is real. I left the company, and 6 months after I left, I got a call from the office of the Inspector General. At which point, Ringer Brothers lawyer talked to him. He said, let me talk to them first to make sure they're not going to go after me too. And they didn't. And so I talked to them. Things didn't make sense to the company. They never did. Turned out the CEO had been embezzling from that $100 million federal contract. Those 2 studies we did, it never really made sense how much money we had and the way things went. And the government figured it out. They didn't end up in jail. They ended up paying a lot of the money back. They got some high-priced lawyers. Did what happens. Yeah, they paid about. They actually settled. I think they paid about $5 million.
That's all. That's all.
Exactly. And it came right down. I mean, they were not smart about this. They had us. When you're in a federal contract, the maximum salary you can take from the government, what is it, 250?
About that, yeah.
250. It's the
125, yeah.
225. So let's say the maximum they could pull out was $500,000 a year for federal salary. They had a party time at the new house at Rancho Santa Fe, where the barbecue and the catered lunch was set up over the pool that looked over the tennis court. So we're all sitting. We're not reading the room. Meanwhile, all of us peons, working away and not getting it, anything. We're, not suitably impressed. We were a little upset. So, note here: don't steal from the federal government. Also, make sure you have a good timeline for what you're doing. So this is another interesting one. This is a right time, right place to. There's a company called Haberle, and I didn't work for them. I like what they did. This is when I was at Baxter. And so when I was at Baxter, I was the clinical lead for the drug called alpha-1 antitrypsin. So alpha-1 antitrypsin deficiency is a rare disease. It's a mutation. Certain individuals don't make it, or they make it incorrectly in a way that doesn't get properly extruded. Your lungs are sort of an area where there's a lot of trickling. You have neutrophils. Neutrophils produce something called neutrophil elastase, which looks to digest tissue. You also make something called alpha-1 antitrypsin, which counters that digestion. So if you don't have that, every time your neutrophils start to get active in your lungs, they digest a little of it, and a little bit more, and a little bit more. And by the time you're 35 or so, you start to get some COPD. By the time you're 40, you start to lose your lung gas, and it goes on from there. Now, this is what's called a plasma-derived therapy. There are certain classes of drugs that aren't made. Like a lot of, recombinant drugs are made in mammalian cell vectors or, plasma-derived or, yeast recombinant systems or even bacterial systems. There are certain drugs that you just can't make that it's better off in the herd of people. And so there are a couple companies that do this. They're making drugs called IVIG. When you have primary immune deficiency, you don't make antibodies. So what do you do about that? Once a week, you go get your antibodies, and you get your antibodies from the herd. And so they have plasma donation centers. Volunteers go in, they take their plasma out, they pay them for that, and they use that to make a drug. You can't make IVIG in any other way. Now, because IVIG, and this is really when it comes to cost metrics, I just think it's an interesting story, but IVIG accounts for 90% of the cost of plasma therapeutics, which leaves you with a bunch of other things, which are basically free profit. And this drug called alpha-1 antitrypsin, which is the second thing. I think it's frac 4-1. So it's, plasma therapeutics is kind of like oil. You have your jet fuel, kerosene, gasoline, different things on the fractionation column. They fractionate in the same way, but called that process. And frac 4-1 is alpha-1 antitrypsin. And because IVIG takes care of 90% of the cost, it's basically cheap profit. And so there really is no reason for anyone else to do a recombinant alpha-1 antitrypsin. But that doesn't mean people realize that. And so there have been a number of companies that did a recombinant alpha-1 antitrypsin. There's a company called OmniBio that did one that was actually very elegant. It was an FC fragment. But all they wanted to do was replace alpha-1 antitrypsin. So it went along like this for a while. There are also some things that very few people know because never published. Alpha-1 antitrypsin can't be given sub-Q. And you would only know this if you worked for Blackstone, which I did. And so they tried different things. They said, okay, we're gonna try this, recombinant drug from a sub-Q. Use that in the value proposition. Well, they talked to somebody who worked for Baxter, they know they want that. And so they wasted much money. And so it kind of chugged until this company difference. And they did a alpha-1 AATFC fragment, which honestly was no different than OmniBio's, but at that point, OmniBio had since gone out of business. Their patent estate had gone up, and that allowed Inhibrx to pretty much seize the entire program. Now, what did they do though? How did they change the story? They actually looked into this and said, we can give this subcutaneous because it's a FC fragment. So what's your value proposition? An alpha-1 antitrypsin patient for the rest of their life has to have, we used to joke about being the infusion experience because it was literally an hour strapped to a chair with an IV because you'd be getting 5 grams of product. And we used to joke, we could give, we give patients a free Netflix subscription with every purchase. But how do you get away from it? What they figured out with their FC fragment is they could do it subcutaneously, and they got 3 to 4 weeks, really more like 4 weeks out of it. So now instead of having a patient who, is trying to live their life, work a job, one day a week, they have an entire afternoon where they have to go sit in a chair and get an infusion. And after that infusion, you don't feel very good. It's called infusion rate reactions. Doesn't matter, doesn't matter what the drug is. If you infuse over a certain amount of protein into the system in a very short period of time, you're going to get fevers, you're going to get a rash, you're going to get a headache, you're going to get symptoms. And it's just going to happen. You can give certain things that help it. Antihistamines help, Tylenol helps, but only does so much. So they came up with the value proposition that from a patient experience perspective, we can do the same thing with a monthly subcutaneous injection. So they have the story. They've got, once again, talk about that actual marker. They had the same marker that they could use, which was, in this case, neutrophil elastase inhibition. So they had a plausible story biologically. And then in January 2024, Sanofi picked it up for $2.2 billion. They didn't do that much. If you think about it, what did they really do? They sat on it, they told the right story. They didn't do a study like Repatha did. They did a study called Elevate. It was, I think, 40 patients. So what did they really spend? Maybe $100 million total on this. And they exited for $2.2 billion. And by the way, this was not a group that needed the money. We were 4 friends had already, four individuals who had already made it in other companies, came together, started their own company, got some venture capital, went around to different universities, looked for basically a free item package, patent estate, found 3 different items, 2 oncology products, the alpha products. So they sold to Sanofi for $2.2 billion. In that deal, they relicensed back their other 2 assets. So Sanofi basically took Inhibrx for $2.2 billion, gave them the registered assets, gave them their structure. They moved on. That's not a bad day for these 4 founders. I think they're, the one I know is they involve nonprofit organizations. So lessons, engineer it, measure it, package. Let's focus on the packaging. How do you tell the appropriate story? How do you tell a story that's going to really get you noticed? What's the frame the problem statement. And move ahead. If you can't adequately frame the problem statement in a financial metric, in a way that says, how does it improve the patient's life? Even when it's the end, even if you're only at the beginning, even if you're just looking at cell lines of candidates, you need to frame that problem statement, frame that financial metric and see where it goes. So, last example, another fun one. Failures are more fun in a way. So this is a company called Aseragen. I was actually the VP and head of early development at Aseragen. And Aseragen had a couple products. The one I'm going to talk about here is, it's called 801, but it's recombinant human acid ceramidase. So acid ceramidase is, basically something that you produce. It's involved in lysosomal storage. There are individuals out there who do not have proper acid ceramidase. They have mutations in it. And it's called Farber disease. Farber disease is a pretty horrible disease. It's lysosomal storage disorder with spinal muscular atrophy. There are different levels of the disease and how badly it affects these kids. If it affects you seriously, you're not making it to adult. And it's not a fun one. They get nodules all over them. So one of your endpoints, potential clinical trial, is measuring these nodules and decreasing them. But even when we were talking to experts in the field, they would say Farber, and they would say to you, you mean Fabry? No, I actually mean Farber. Because Fabry is also rare disease. But Farber is so ultra rare that even like pediatric rheumatologists will move it. And so you're talking about a couple thousand globally, maybe. But then how do you find them? And so in this case, the Aseragen actually launched by acquiring this Enzidant. So Enzidant was, on the bats, and they, look for, old patent estates that they could bring in. They never went anywhere. It's probably because they figured that it was a very important. Because they have very smart people. But they got money. And so in this case, it was 35 million. That was something called product financing. And so product financing is interesting because it's not an ownership stake. You're either taking a royalty. So NovaQuest, which is a interesting group, they came out, they're the old venture capital arm Quintiles that now is standalone. And they don't do ownership. So like normally in a venture capital, you give up, let's say 20%, and you value the company, and let's say 10, easy math, they value the company million, they take 20%. That's not what NovaQuest does. NovaQuest says, let's make it more complicated. We're not going to take ownership. But because they don't have ownership, they don't have as much of a stake in the game. So in this case, this goes back to that priority review voucher. Because this was a pediatric rare disease, it was eligible for priority review voucher. So all Aseragen had to do was get the product over the line. NovaQuest never cared they sold it to anybody. They just cared that they actually filed it, got the product over the line. They would get the entire priority review voucher, which, as I mentioned, goes anywhere from 60 to 300 million depending on the time. And it's venture capital calculations that we use 80 to 90. And so it's a good investment, right? Give up 35 million. Talk about what we talked about earlier, which is markers, right? The population markers. Simple enzyme replacement therapy. These patients don't make acid ceramidase. You give them acid ceramidase. They have acid ceramidase. They do better. So it's a very simple story. But now we start to talk about ultra rare diseases and the patient front. Instead of can you find the patients? Let's also talk about the fact that the severe patients die very early. So it's even harder. So this idea that you have 2,000 globally, now go out and find them. And maybe it's not 2,000 because the more severe ones are dying early. Now let's talk about your clinical trial, right? You can find the less severe ones because they live to adulthood, but the less severe ones don't have the severe disease to make them that much better. So now you have a harder time. I talked about those nodules. You might have cancer, but maybe you don't have a lot of nodules because you have less severe disease. So now how do I have an endpoint of nodules when I have a patient who doesn't have them? And so I have a more difficult time running my statistics. So this kind of all gave into a friction point. Spend a lot of money trying to find this. Patients through all kinds of vendors to tell you to find it. They can't. But they'll tell you they can. They'll sell you on jargon. You start working with all these EMR vendors. You start working with genetic repository vendors. Just don't matter. They're not out there. And so the problem is that the early stage people, when they make their pitch to venture capital, use very rosy data. And in this case, they cherry picked some publications. We said they're buying about 3,000-4,000 of these cases worldwide. We looked. We found 100. I mean, we really looked. So we did all we could. And of those 100, unfortunately, 25 ones we found were deceased. And so that means 75. Now, of the 75, maybe 50 of them were lower disease, severity. So they were 35 to 50, actually a wife. But they didn't have the symptoms. Yeah, they had the diagnosis. They weren't perfect, but they didn't have the same kind of symptoms that you could show a real endpoint in the trial. So again, you don't have a good patient finding plan. You don't have a central scientific strategy. You don't have the operational ability to execute. And that's what people are looking for. Can you execute? This is very unique because Nova Plus didn't really care if you could execute because they figured you could, because all you needed, for these ultra rare diseases with FDA, you can file on 12 patients. I mean, how are you going to file on more? And so we probably could have gotten them. Unfortunately, there's also financial mismanagement of the company, and that's at least one. I realized this when all the VP involved, we were all sitting at Pinehurst Golf Course in North Carolina for our leadership retreat, where we played 9 holes a day, minimum. Had a nice dinner, smoked some cigars, drank a bunch of whiskey. I don't know, talked about the future of the company for half an hour. And and I'm sitting there thinking, for all the money we're spending, I could take this drug, and I had some other ideas for the drug, still do. I could take this drug, run an animal study in this population, or something that I think actually has everything we're talking about. It has a diagnosable population. It has, a payer response. It has an easy to run clinical trial, and there's no patent estate in the area. So there were some ideas I had that, for the same amount of money we gave for that weekend, and not weekend, week, 5 days in Pinehurst. And it is a very nice golf course, I will say that. It's a fantastic place, as long as you're not paying for it. But yeah, I could have probably gotten us another patent. That kind of one. So it's a little, opportunity for us. And there we have, yeah. Investor wants money back as Durham fee. And that was NovaQuest wanting their money back. And the company in Wayback, so the company went to go to that. So it's another trip. It's something called ABC, which is Assignment for Veteran Creditors. ABC is basically bankrupt, bankruptcy proceedings, but not through a judge, through an agreed upon lawyer. And so what it does is it allows the CEO of the company who messed everything up to not have a bankruptcy on of the company. And so it's all private. The problem with that is, as the employee, you don't even get your severance. You get listed as part of the creditors. So all of us in a fun Friday at 2:30 meeting were out of nowhere, all furloughed. And this includes my buddy, I was VP head of road development, my buddy, the chief medical officer. We all learned about this Friday at 2:30 p.m. The CMO didn't even know this was coming. And we had another contract with a different product with the Army, DOD, for an antibiotic. I think as they put it, how are you guys going bankrupt? We're paying you money. We had a $50 million contract with DOD for medical products. The extent to which you have to mismanage your company to go bankrupt when you have a $50 million DOD contract is honestly staggering. And so what they did. This product, yes, it's ceramidase. It's got other uses that are already patented that we had patented. One of which is it tends to preserve organ function in vitro. And so think about the nation's supply of organs, right? When you have a donor for a kidney, we're way low on kidney donation. Donations are ranked ABC. C's are not fit. If you had a product, and this product potentially could do this, there was patents on it, there was data on it. It tended to preserve the oxygenation and decrease apoptosis in vitro in organs that were being prepped for transplant. We could have done that. There's so many things we could have done that we weren't able to do because of this fashion. So it's kind of a story about de-risking. It's a story about one, finding the right patient population, knowing when you shouldn't be doing something, and stopping. You know, one of the best things you can do with an early stage program is try and kill it. And if you can't kill it, it's an entirely different way of thinking. Try and kill your program. If you can't kill it, you've probably had success. Throw it into some studies that have a very difficult chance of succeeding, but you do look at pushing forward. Don't get too attached to it. And that's always a problem with early stage startups. That's one of the things that big companies do very well. As you know, like when I was at Amgen, I was on a drug called 986 for a while. It's a cardiovascular drug. Never went anywhere. They killed it. I went on to a different program. They didn't lay anybody off. Nobody lost a cent of bonus. You knew that going in there. So you weren't worried about killing the program. You wanted to do the right thing for it. If it wasn't going to go anywhere, you're going to kill it. Now, it doesn't always work that way. I mean, Amgen acquired a drug called Tavneos about 3 years ago. Tavneos is in my opinion, a horribly lousy drug. It replaces corticosteroids as a slightly less disgusting adverse event profile. Steroids are rough on patients. Decreased bone density, glaucoma. They're very rough on patients. Weight gain. But they're the only thing out there for a lot of autoimmune patients. So having something else, that was a C3 complement receptor inactivator in this case, having something else made a great story. But the clinical trials were rough. They were horribly done. And this comes, sometimes pharma just has a check they have to spend. They have to spend money to make it look like they're bringing these things up. And so they end up acquiring Tavneos. I think it was like $3.74 billion or something. Tavneos is sold basically nothing because it's a lousy drug. More recently, some of its liver effects come, and the FDA actually asked the Amgen to pull off the market. So all this stuff about, big pharma's making great decisions. No, it's great decisions. It's just a horrible decision. Spent $4 billion on it. Now the FDA is requesting they pull it from the market. And so, it all goes back to there's a lot of uncertainty. So de-risk as much as you can. Try and kill it. Don't be worried about the data as much. Don't try and, if you find yourself, let's put it this way, if you find yourself working for a company and you think that management is so in love with their drug that they're not seeing it clearly, that's a pretty good sign. Listen to the actions, don't listen to the words. Look at the data. And sometimes it's time to move on. You look at people who've been at pharma for a while. I've been with companies for for about a year and a half. I was with another company, I was with Vertex most recently. Because of, agreements, I'll just say I didn't enjoy it. I was there for 2 years. Moved on. Vertex is interesting place. They potentially have one of the best medicinal chemistry groups on the planet. They're the only group is set. The drug we worked on. And Shankar knows the drug that I was on. It's called APOL1, mediated kidney disease. And so APOL1, and actually we're working with Harry on this. APOL1 mediated kidney disease is really interesting because in a way it's a correlate story of the malaria story. Everyone knows the malaria story very well, right? If you have sickle cell, if you're from certain regions of Central Africa, if you have sickle cell, you are partially immune or immune to malaria. Sickle cell trait. So if you have full sickle cell, your life is not easy. It's pain. You have a lot of issues. Now, for trypanosomes, there's something very similar called APOL. If you have APOL1 heterozygote, you are immune to African sleeping sickness. So if you're in Central Africa, that's a very good thing to have. If you have 2 copies of it, there's a good chance your kidneys are going to start to fail early on. And so throughout a lot of history, and this is absolutely unfortunate, African Americans were looked down upon for having increased rates of kidney disease. It's their lifestyle. They're not controlling their blood pressure. They're not this one. There's a genetic reason. African Americans, especially if you are from, Nigeria, Ghana, that area, there's a very good chance, I'll just say this right here, why get tested? If your lineage is from Ghana, Nigeria, it's free. There's a number of places that physician can get tested. No reason not to. Especially because that drug's a great drug and it's probably going to be out on market in another 2.5 years, even less. But this goes back to the story. Once you have a good drug, and it's a great drug. There's something called uroprotein cracking ratio. Basically, it's a measurement of how leaky your kidneys are. We saw a 65% decrease. This is published in the journal medicine. We saw a 65% decrease in the amount of protein being spilled from the kidneys of APOL1 positive high-risk patients with kidney disease. That's a beautiful. It's currently in a phase 3 outcomes trial. We had over 250 sites globally. Like I said, go where the patients were. We had 5 sites in Nigeria, 5 sites in Ghana. I spent a couple of weeks in Nigeria with all the sites. It was an incredible experience. You go where the patients are. You have things, that you do what it takes to get things done. The average, if you look at individuals of African descent, Central African descent, who have kidney disease, and then just enroll them in the trial or start to screen them, we were running about 10% inclusion. So only about 10% of individuals, we tested actually had high risk genotype. And that's because, African Americans in the United States are from all over. They're not all from that Central African region. We had a number of interesting observations down in Brazil. We actually could follow some initial slave routes down the coast of where people had gotten settled. And that's where a lot of our clinical sites that came, individuals who came out of Central Africa. But I said 10%. We went to Nigeria, only 40%. And so we put the patients. Nigeria, we had horrible enrollment. Year of dying. Going to Ghana and Nigeria saved the trial. The trial right now, you can look it up. We actually closed the initial enrollment. Or not we anymore, because I'm not there anymore, but they did that. And so it's a fascinating go. You do what you have to do. A lot of those sites we had in Nigeria had never done trials before. Things popped up that you wouldn't think of, like the fact that there's no electricity for a couple hours a day. So we bought solar generators, bought battery systems, had them installed. Trained, we had a big meeting here in the US. We brought all of them over here, did full training. You do what you have to do to get it done. I think that's one of the lessons of drug development. Do what you have to do to get things done. And so we talked about these different trajectories, let's separate. Honestly, the pro-inaxaplin, which was the Vertex APOL1 kinase program, that falls in the way upper right-hand corner. That is a great program. That is a great marker. There's great data. The chemistry is beautiful. The patients, as I said, they were originally tough to find. But as time goes on, it got more and more. There's a lot of work we're doing behind the scenes now, or even my friends still at Vertex doing behind the scenes to try and get this genetic test on the newborn screening panel. Just like cystic fibrosis, it's on the newborn screening. When you're born, if your child's born with cystic fibrosis, there's no reason you should not know if your child is a APOL1 high risk kid, because you would do different things. So a couple principles. Start with the end in mind, but choose the next decision first. Kind of take that last decision, bring it forward. And everything you do, you have to be very blatant in what you do. What's the next decision? What's this study going to decide? Don't lie to yourself. That's the big one. What must be true? What would justify the next step? And the other thing about what's the smallest study that can answer it? Cheap out when it's early on if you can. Don't fall into this trap. Don't go to Pinehurst. Basically, find a collaborator. Talk to some people at Vanderbilt. See if you can't find a professor who's got interest in this that we can collaborate. Look around internally in the Harry. Do you have a professor here who has patients that have biopsies that maybe, what's your marker? What do you look for? And in terms of your action, set things up in advance, set up your milestones, set up your go, no go decisions. And if you hit no go, listen to the actions. There's nothing wrong with it, no go. Things our programs out there for questions. Sometimes you have to know when to stop throwing good money at. So talk about this a little bit. Back to the general concept of biomarkers, dual use biomarkers. Some biomarkers support your FDA decision, LDLC. Other biomarkers maybe just get you additional money from venture capital. There's got to be a reason why you do everything. That reason can be, I want more money. There's nothing wrong with that. You have, I don't know, maybe you've got an SBIR, and you know that SBIR ends 2 years. So you have 2 years to figure out what a package can be that will get you the $10 million seed round for venture capital. So think about that. Where do you do that? What's your threshold? Functional alpha-1 antitrypsin, that's that inhibitors thing. They had a marker for it, but that alone wasn't enough. They had to show something more. They had to show they could do something that no one else could do. LDLC with Amgen, with Apple, that was enough. Now, treat feasibility like translational science. Do you have the patient population? If you don't have it, if they don't exist, you can't go farther than that. You've got certain burdens. When you take money from a venture capital group, first of all, it's nothing. But even my company right now, I'm not, I don't want to talk about my company right now for legal reasons. But we meet with our board all the time. And that's not like me, loving the board. We meet with the board all the time. They want output all the time. It's not like, it's not a quick slot. It's a hard work. And it's the latest thing to do. So make sure when you're taking that money, you've got your cadence, you've got, right, is make sure they've gone into it. Reduce the burden as much as you can on different things. I always talk about patient burden when you're writing a protocol. You always have to think, when you're bringing actual patients in, have sympathy, recognize what they're telling you, do some work on their disease, talk to some patients, know what they're going through. If you're doing a disease and alpha-1 antitrypsin deficiency, as I mentioned earlier, those patients have COPD. When they get advanced, it's hard to get around. Are they parking on,
Recording in progress.
Are they parking at Meharry and trying to find this building? That's the 6-minute walk. So if you've got your clinical trial site in this building and it's a little rough. So think about what you're putting them through. Look at your blood volumes, the little things. Don't exsanguinate your patient. Because it's easy, especially when you get a lot of early development people. I want to know. I have 700 different tests I want to run. I want to know all these antibodies. I want to know every single cytokine. I read about this once in research. I want to see if this happens in patients. No, don't do that to your patient. Keep it simple. Think about what is the actual information you need to get to your next delivery. What's your next point? What's your next decision matrix? Get through your gate. And think about partnering. Always recognize where you're going. We do a lot here with Life Science Tennessee and SBIRs. Federal government's not important. They're a great way to move forward, right? Non-dilutive finance. Someone's going to give you money. That's beautiful. But they're not your final partner. Their money runs out, and you have to be able to take it past them. At some point in time, you've got to convince someone that there's profit behind this. Someone who, quite honestly, is not a great person, is not a nice person, doesn't like you. You'll be lucky to get in, even get in with them. Now, I was on the phone with a high-level partner in venture capital group, and it was a Zoom call. One of the first things he did in the call was, basically made some jackass comment about me being in my basement. you know, my office, my basement wall, he's in San Francisco, so I have to do it. Being a bit of a jerk myself, I respond to him, I'll have you know, I'm actually in, you know, over the garage. So. But they're not nice people. They're in it for themselves, you know, if they can, they will push it to 51%, push you out. Keep that in mind. You know, good packages early, make life easier later, put the work in. And so tell me a little bit, you know, what can you do for a 90-day startup? This is not actually doing it. This is kind of, you know, a 90-day startup of what you want to do. Studies obviously take long. You can't even contract a study. But look into this early on. Define your decisions, define your mechanism. Look at that patient population. What's the unmet medical need? Start thinking about how much you can charge for this. I know it's brutal, but it's the way the system works. And people are going to ask you. Start thinking about where it goes, the number of patients you have out. Second part is what assays do you have? What's your marker? LDLC is a beautiful example. You're probably not going to have something so simple. But there are things out there you can write the story. Make it a story. You know, make it interesting. When you're pitching, you're a storyteller. And when these big money people write checks, sometimes they're investing in a person as much as they're investing in an idea. So make them want to invest. Have a good story. Have a good personal story. Have a good story about why you're doing this and bring it forward. You have to have an operational plan. You know, it's got to be vetted out. CROs are good at that, but they will write something up for you. You know, you want to spec out a phase 3 study, they'll spec one out for you and give you a quick proposal at full costs. Those costs are probably going to freak you out, but it doesn't matter. They're real. You put those in the deck. Same thing with manufacturing. You know, a lot of people stop in. There are additional things manufacturing. Your regulatory costs. It'll cost $100,000 to file an IND. That's what doing most work yourself. You need the cheapest vendor out. Go to a big vendor, it'll hit $1 million. But I don't know. These are all things you have to do. But the cost, being realistic, if you present to a venture capital group and you're not realistic in your costs, it's a massive first sign that you don't know what you're doing. And they could love your idea, but they're still not passing. They don't believe it. So have some logic to your dose, you know, PK, PD, PK pharmacokinetic, PD pharmacodynamic. It's how do your drug levels affect your marker? Where does it go from there? You know, there's certain things you're not going to have in your early stage. You're not going to have safety. Okay. That's fine. But you have to tell a story. You have to have a regulatory story. You have to have a path. You have to know where you're going. And so that first study, this is the last slide. First study should buy your next 3 decisions. So going back to that hardcore science, but explainable science, financing, and your eventual out FDA or partner. Doesn't have to be FDA. Okay? Getting phase 2 proof of concept, the idea of selling a compound is a very, very valid strategy. Doing a big FDA phase 3 and getting registration. Less people want you to do that. Let the big company handle it further down the road. Do proof of concept. Show somebody what you can do. And that's it. Hope you enjoyed it.
Questions?
Next one. Yeah. Thank you for the very nice talk. Very interesting here. I'm actually a computational modeler. So I was wondering, how does this work for the software, the computational models that can be used for diagnosis and also for individual?
So that's a great question.
Any experience
in
in
the
that?
age of AI right now?
Yeah.
It's a perfect question because there are so many companies out there that are basically coming up as a service now, and they're being launched. So the ideas are being, so right now it's actually a very hot area, and money is actually not always easy, but easier. And so you still, the same modalities apply. It's just your computational model starts your student. Now the question is, how do you show that your computational model works on the real world. How cheaply can you do it? How quickly can you do it? And that's when you go, so what do you want to do with it? The eventual goal of everything is to license it out, right? I mean, we can all say that. You know, nobody's looking to be poor. We put all everybody. So the computational model is a great start. It's your story, it's your foundation. The other thing is if you use computational models, you've got a different idea than what happens. But to show that your model is valid, take something that's existing out, back calculate model, and show that your model came out exactly. It's that. I don't know if it's a diagnostic, oncology diagnostic, or oncology product, or something of that nature. But the more data, the more specific these examples, the better off you are to actually base it.
Yeah, but listen, I really love the stories. I was curious listening to you this politics in terms of decision makers at big pharma versus small companies and how they interact with the clinical development side.
That's a great question. I'll go back to the original slide I had, which said the answer is it depends. And so I can tell you that some are very science-based, others are very personality-based. Certain company I worked at recently. Not gonna, yeah, you want to say the name, but everybody knows which company it's in, so. It has 3 people. The entire business development group there is a bunch of bankers. They don't make any decisions. The top 3 people at the company decide what's going to happen. And they tell the bankers to go make the deal happen. And that's it. Then you go back to Amgen. So Amgen was really very science-based, great place to work. And when you're in clin dev at Amgen, you get lent out for different things. And so one of the most fun times I had was I got lent out a couple times on due diligence teams. So I was part of the group that actually looked at external assets to bring in, and which gave me a great window into, you know, what people look for. And that's the thing where you're literally there to shoot down the science. You're literally there to, you know, think, how can I kill this? And they actually even tell you that. So that's the mentality of Amgen. How can I kill this? And how can I make it, you know, how can I put a bullet in this idea that someone else is bringing in? So we can, a couple of reasons. 1, not make a mistake. 2, list the reasons why it's horrible so I can push down the price of acquisition. And then move it in. Now, that all makes a great story. And that's what happens a lot of time. But then other times, when I was there, there's something called this in the cholesterol field called CTEP, cholesterol transport esterase inhibitor protein. And at the time, none of us actually thought CTEPs were going to work. I mean, there were bunch of studies. It just didn't make sense to us on biology. The decrease in the LDL was really more of an artifact because it had to go it basically, you know, PCSK9s remove cholesterol from the system. Statins remove cholesterol from the system. CTEP transfers cholesterols to different areas. So it artificially decreases your LDL while increasing the cholesterol. So we never thought it was really good work. So none of us believed in it. And we bought one for $360 million anyway. So everything they tell you. So I guess that's part of the answer. Everything is talking about Haberle. That company was a guy named John Castelline, who we knew was a famous cardiologist. And after it didn't work, he actually bought the act. So he took $360 million. Amgen shelved the asset, then he bought the asset back. But for some sometimes you can pay them. And then small companies, it's also there's because you're like a fashion thing.
So it's a fundamental question. So you mentioned that there are like 48 drugs you have tried, I mean, with Schrodinger, right?
Yeah.
And none of them worked.
So the idea behind this was neutrophil serine, you know, this was a series of neutrophil serine protease inhibitors. that I was working with a group on. We went to a group and used a computational system called Schroeder, where we, in situ developed over 500 theoretical topics, full structure, predictive analytics, predictive finding coefficients, blood-brain barrier. And so at the end of it, I mean, it depends how much money you have. Sometimes you can, do all 500, but you have to pick, certain ones to go on. 48 of those went to that. Sometimes formulation is difficult. Sometimes, your structures that you think of, you just can't make them. And that's kind of what happened. Out of those 48 that went in, only 10 of them were even, able to be made in sufficient quantity to test. Those 10 that we pushed out, I mean, they were ugly looking compounds. They were not like some beautiful white powder. One of them was brown. Another one was like this pasty lipophilic oil, because you could only like have it in a certain oil substrate. So none of it even looked. So then we set it to a functional assay to see if it worked. And none of it worked in functional assay. So that's kind of that, we talked a little bit, when does the computational validity not mean anything at all? And if you talk to venture capital companies, that's a great question. Talk to venture capital companies, they've seen enough of these that they're just going to pass until you get to the point where you can actually synthesize it in quantity and show it in a functional assay. And at that point, you go back and start thinking about BAC bands. So before that, you're looking at SBIRs, because then you can tell a story and do it under an SBIR, or angel investors. As I said, there's nothing angelic about angel investors. Or you just hit up your friends and family and, see if there are people like, people who will invest some money in your fledgling company to take it from them.
So is it like the something to do with model assumptions? Is the inability to produce it correctly? Which one is?
And the answer is who knows? And so because some of the model assumptions for this case were based on actual molecules that we knew how they performed, that were done in the academic setting, but had no package structure around them. And so we knew there were existing molecules out there that had certain structure that did what we wanted to do. We had a lot of publications out of that. The problem with this one is the professor who created all of them never packaged it. He just published them all. And so what do you do with that? The answer is you can't do anything with it because you can't, raise money on it. Now that professor had, patented anything at all that they did, we would have been on the phone with the tech transfer group at university and just, in-license them. And it would have been a beautiful thing. We probably actually would have been there was enough data in the publications on what the professor had done that we probably could have gone straight to CBRN. We never patented it. And so we had that as model. We had certain performance metrics that a certain molecules as a model that we could use. Still in that. Because we had to engineer around the patent. So we took the patent, or engineer a patent around the existing, literature to make it quote unquote not obvious. So the patent has to be not obvious. Okay.
Question. That molecule you keep speaking of index.
Never did.
I'm wondering why there's the benchmark, it's the engineering first and then the benchmark. Why aren't those switched? Because IVIG is very, very cheap.
Yeah.
And you can in the first studies that learn, why it's almost like you're building bias into.
So the real thing about it is, the real thing on the map is it's all about patient experience.
Okay.
And it's just like,
So that's okay.
yes, that's really just about patient. What can you do? It's just like, what can you do to improve upon the existing? Right now, with all these hospital therapeutics, you've got a patient literally 1 day a week having to go to an infusion center or sit in a chair for multiple hours and, having a pretty miserable experience. Now, if we can give them, even once a week, once a month, either one works if you were using an auto injector and going straight sub-Q. So in this case, instead of going and sitting in a chair for 3 hours and having your day ruined, you go to the fridge, pull out the fridge, leave it on the counter for 30 minutes, grab a chunk of your stomach and stab yourself and you're done. You go about your day. And so that's the type of thing. It's what's patient preference. Every patient out there's going to want to do that instead. So that's your value proposition. Yeah. And so basically you're showing equivalency scientifically Then you're showing how much more money, how much, how
I
much kind of money you can make
I see.
based upon patient preference.
And that's interesting to his question too. I'm out of the industry for like about a year, but then financing and then trying to make the decisions and like when you're in the actual lab and you're trying to fight the money people. It's like, oh goodness. So, thank you so much.
Would you want to say?
So just before we close, so I like to express my gratitude to all of you for coming to Meheri and my Meherians who are here for asking questions and also for visiting us and giving this thoughtful and nice, words of wisdom that I think are very much applicable to the process of discovery and bringing the solutions to market. And thank you. Maybe I see some good faces today and also on the Zoom, we had some good friends from Vanderbilt and some of my colleagues. So thanks to everyone.
But it's not, you know, better.